Hey, good morning. So today we will go ahead and review for our first exam, which is coming up this Friday. Um, so a couple quick reminders. This week you also have a homework assignment due Friday night. Um, I encourage you to try to finish the homework assignment prior to the exam, because I think, of course, going through the process of doing the homework will sort of help you think about some of the topics that will show up on the exam. And also that way, when the exam's over, you don't have to still remember that you have to, turn in the homework, right? So I encourage you to try to get it done before the exam on Friday, but technically the homework is not due until Friday night. In addition, you do also have your weekly quiz. So that weekly quiz, I think I told you that I'm giving you a little bit extra time on it. I think it's due tomorrow night by midnight. I also encourage you to take it as soon as you can, because also that quiz will help you review as well. And I've set up the quiz so that when you submit your quiz, you instantly get access to the answer key to help you study. Okay. So again, you have until Thursday night to turn in the exam the quiz, because I know you're busy trying to figure out how to study and pace yourself now. But I also encourage you to try to get that done before Friday, or as early as possible, so that you can really take advantage of it. All right. So again, we looked at this last time, but just to remind you that the format of the exam will be 25 multiple choice. You pick the single best answer, A, B, C, D, and then you will be given 4 topics that you can pick 3 of them to write 3 short answer responses. And then you'll be given 2 topics and you pick one of them to write a longer response. So the 3 short answer ones, typically it's like maybe 5 sentences or so. You can provide a adequate answer to get full credit. For the long answer one, it's more like a paragraph, right? Because it's a little bit more complex. And so you will have the 50-minute class time in order to take it, right? So Friday morning at 10:30, log in on Zoom with your webcam working. Remember, I reminded you that if you're having issues with your webcam, you can always use your cell phone to handle the Zoom part of it, so that myself and the TIs can see you. And then when everybody's ready to go, we will open up the exam, and then you can take it in La Lima at the same time, right? Um, Oh yeah, right. So here down at the bottom, I mentioned that the quiz 4 is due Thursday night. I always like to remind you guys about these things, especially when things are due at a different time than they normally are, because, if it's me, stuff that's outside of the routine always throws me off, right? And I would be the student who forgets to do it. Okay, so that's why I want to remind you. All right. So today's review, we have about 20 slides to go over. I always say there's no way that I can go over every lecture in every detail, but what we'll do is hit upon some of the key slides as examples from different lectures, to help emphasize that there are certain topics that are really important. And hopefully today's review session will help you kind of wrap your head around kind of like a strategy of what topics you want to focus on as you pace yourself to prepare for the exam. Right. So we started off this semester talking about how tumors are very difficult to classify, because tumors come from potentially any nucleated cell in our body as the starting normal cell, right? That means there is a multitude of different possible tumor phenotypes. And so we jumped right in at the beginning of the semester and started looking at how challenging it is for oncologists, especially clinical pathologists who are also oncologists, to try to understand what they're looking at when they're looking at a patient biopsy. So go back and review that there are 3 main ways of categorizing tumors, either by differentiation state. In other words, where these cells most likely originated from. Also, the embryonic origin. Remember, at the early development of the embryo, when you were just basically like a volleyball or a basketball made up of cells, hollow sphere of cells. At a certain point, these cells begin to differentiate and start turning into the three primal germ layers that give rise to all the tissues in our body: the ectoderm, the endoderm, and the mesoderm, right? And that's important because during those early steps of development, these will be like the first steps where certain sets of genes are now turning on or turning off differently in certain cells compared to others, right? And that sets the stage later on for the fully differentiated tissues to have a very different sort of original set of differentiation steps that gave rise to those cells. And third is biological behavior. Right, that one's kind of simpler to remember. Right, either something is benign. Right, so remember, you can have tumors that don't turn into cancer. Right, just because you have an overgrowth of cells, that does not mean it's cancer. Right, it's only when those cells undergo additional changes that give rise to malignancy does it become cancer. Right? And we will look at those in more detail later in the semester. But for now, it's important that you're comfortable with that idea, right? That not all tumors are destined to become cancers. Although another way of thinking about it, though, is that, of course, the formation of a tumor certainly is part of a cell transforming all the way to becoming cancer later. Right? So early examples of tissue that we looked at in the semester: epithelial cells are particularly important for our class because I think you guys are already aware of this. There are so many different types of cancers because of the diverse multitude of different original starting cells that they could give rise to, that we tend to focus in our class just on epithelial-derived cancers, right? After all, epithelial-derived cancers represent like 80% of the types of tumors or the types of cancers that are diagnosed in Western clinics. So that's a good justification to focus on that. It's another good reason to focus on epithelial-derived cancers because they serve as a good model. Many of the changes that take place in epithelial cells as they go through transformation and eventually become malignant, right? If we study those models or steps in the epithelial cells, it's a good, convenient way to also learn and apply them to other types of cancers that we might want to study later on. So review some of the features of epithelial cells. Remember, epithelial cells, they are attached to a basement membrane, as shown here, underneath which you have the mesenchymal cells, right? Or sometimes referred to this as the stromal cells. This is more like connective tissue. These two illustrations, I think, were a good introduction to that. Recall that we have certain nomenclature or naming conventions for things like tumors compared to full-blown cancers. Remember that omas, right? The suffix oma typically refers to the formation or detection of a tumor. And if that really becomes cancer, we refer to them as carcinomas, right? And in particular, since we're talking about epithelial cells, right, carcinomas are mainly used as labels for different types of epithelial cancers. If they came from glandular epithelium, which is quite often the case, that's when we call that an adenocarcinoma, right? So adeno denotes that this was glandular cell in origin. Go back and look at some of the early slides that we looked at where we were beginning to appreciate some of the obvious phenotypic changes as cells go from normal to being cancer, right? Things like loss of proliferation control, loss of proper morphology, right? Organization of the tissue becomes disrupted, and not just the epithelial cells themselves, but there are consequences on the connective tissue that support them, right? So think about those things and how those types of changes, are kind of present when you look at these different photos in the lecture slides. So this is an important conceptual figure because it helps us understand, right, that cancer cells are cells that have improperly shifted their focus. Right? Fully differentiated cells, for example, this differentiated cell type 1, this green one, or this fully differentiated cell type 2, right? They, of course, originated from, if you go far enough back, some stem cell. Right? Here we have this labeled as a pluripotent cell, so this would be really far back, maybe as far back as embryonic development, but maybe for practical reasons, we can actually say, let's start with a multipotent cell, something like our hematopoietic stem cells, right? Cells that have differentiated enough, right, to be able to be specific to a particular tissue type. For example, the hematopoietic stem cells that are now in your bone marrow that can only give rise, right, to a selection of other blood-related cells. Right? So a hematopoietic stem cell can still become one of roughly a dozen different types of fully differentiated blood cells, whether you're talking about white blood cells or red blood cells or platelet forming cells. But importantly, that multipotent hematopoietic stem cell is far enough down the commitment sort of flow chart, right, that it cannot become a nerve cell or a muscle cell or some other kind of gland cell, right? So let's start with that. Remember, stem cells are tightly controlled, right, because their main job is to proliferate. As they proliferate, their daughter cells go down additional steps to differentiate and become the cells that you see on the right. And these fully differentiated cells are now concerned just with doing their fully differentiated cell jobs, right? Their tasks. Keep in mind that there are rare instances where related differentiated cells can trans-differentiate, but typically, we don't really talk about those because it's really more the exception than the rule. But if you start getting random mutations that start leading these cells backwards and de-differentiate, right, you start getting into trouble. Right? And so obviously those mutations, if they're random, some of them will be selected for because some of them will actually give these differentiated cells some advantage, right? Other mutations, if it doesn't give them an advantage, then maybe those cells just go away, right? Because they actually have trouble surviving. But the point is they're becoming sort of more stem cell-like again. In other words, they're giving up their differentiated jobs and becoming more preoccupied in a way with this idea of proliferating again, as though they were a stem cell. But the problem is, right, those random mutations are not going to lead them back nice and neatly along the dotted line. Because they are random mutations, they'll lead them astray, right? So you get the screwed up phenotype that unfortunately now, right, would then be proliferating out of control, and the whole right part of the screen would be filled now with this cancer cell type. Right? But again, it reminds you that it's difficult to know what the original differentiated cell was, the normal cell that gave rise to this. So we started our examination of oncogenes by appreciating the fact that quite a few types of cancer are linked to viral infection. Remember, about 20% of cancer types can be linked to viral infection. And I always underline types, right? Because I don't want to get confused and say, out of all the cancer cases out there right now in hospitals or wherever, that 1/5 of them were due to virus infection, right? No, that's the wrong way to think about it. Remember, I said if you were to list all the different types of cancer, you would find that on that list, about 1/5 of them, right, there is evidence that certain viral infections contribute to that type of cancer forming. So these are some examples. HPV linked to cervical and throat cancers, Epstein-Barr virus, and these are pharyngeal cancers, hepatitis B and C viruses linked to hepatocarcinomas, right? And HIV linked to AIDS, which of course is just opening the door for certain types of cancers to actually take hold that normally the body would resist. Right? We examined how DNA viruses integrate as random fragments in the genome, right? And they're messy because DNA genomes of viruses tend to break apart while inside the cell and form these weird concatemers, right? Multiple copies of the DNA genome, and then those fall apart, and little bits and pieces of them integrate all over the place in the genome. And we said that researchers have found that in certain cancers, for example, cervical cancer, right, it's almost a given that when you take a biopsy of cervical cancer tissue and you look, pathologist looks for evidence of HPV fragments in it, right, they'll find HPV fragments in those cancer tumor cells, right, in the genome. And importantly, what? It turns out it tends to be the early region of the viral genomes that integrate, because those are the early region genes that are part of the virus's genome that encourage things like the infected host cell, right, to no longer be satisfied being a differentiated cell in G1. It starts pushing or encourages the cell to move into S phase, where DNA replication happens, right? Because that's what the virus wants. It wants to hijack the DNA replication machinery of the host cell to make more virus particles, right? Normally, remember, if the whole viral genome is intact there, then the late region viral genes take over, and those are the ones that then code for the different parts of the virus particle that the host cell now makes. And then you have a whole bunch of new virus particles made, right? And then the host cell dies in the process, right? And that's key. So in other words, if the virus is successful in hijacking a host cell to carry out its bidding, right, then that host cell dies and you don't get cancer, right? But if something goes wrong and the virus kind of screws up because it doesn't integrate properly, and you get all these messy little bits and pieces of things like the early region genes, right, that might encourage the host cell to start going through the cell cycle without ever successfully making virus particles. And that then, right, continues to force the cell to divide. And that's, the first steps in transformation. Right? Remember, RNA viruses are a bit different. The classic RNA viruses, because their genomes are RNA, they have to first, in the cytoplasm of the infected cell, undergo reverse transcription to make DNA from it. Right? You get these nice, neat, tidy DNA copies of the entire viral genome, right? And these get integrated, typically intact in one piece, and we call that a provirus. Right? And from that provirus, then the hijacked cell starts doing a lot of transcription, and the intact viral RNA then gets put together, right? And that's how you have your RNA pieces. Remember, certain viruses that are RNA based, like coronaviruses, for example, like our SARS-CoV-2, right? They actually are different. They don't go through the integration as a provirus. They just basically start from the fact that they're RNA, and they have certain sequences in their RNA that actually allow the host cell to go ahead and just use that RNA for translation, right? So that's why you would have some RNA viruses that probably will never have any evidence of being linked to cancer, right? So, the SARS-CoV-2 probably will turn out that there will never be, years from now, any evidence that COVID-19 somehow increases the risk of certain types of cancer. As far as this mechanism goes, right? There might be other things yet that we haven't realized about, what physiological changes in the body following COVID infection. Right. So then we began to appreciate that when researchers looked at things like the Rous sarcoma virus and compared it to non-tumorigenic related types of bird viruses like the avian leukosis virus. Remember, Rous sarcoma virus was first described in chickens, right? So clearly it's also some kind of a bird virus. What the researchers found is that the RSV and ALV genomes are strikingly similar, with the exception that the RSV has an extra bit of sequence at the end, right? So this is what they called, oh, the oncogene, right? It must be this thing that's causing the transformation of the cells, right? And they went on to call this V-sarc, right? Because it causes sarcomas in chickens. That's the tumor that they were originally identified in, and it's a viral origin, right? So V-sarc. And then remember, we learned this kind of interest. I think this is very interesting, right? This thought about how does something like a V-sarc gene actually arise? And we learned that most likely it was the result of avian leukosis virus or some other kind of closely related bird virus infecting bird cells. And somewhere along the line, right, it integrated in a site right in front of the sarc gene, right? The normal C-sarc or cellular sarc gene that then kind of got borrowed, if you will, right, became part of the viral genome that was being transcribed. And then later on, as part of the virus particle, right, the sarc sequence got mutations in it, right? So that's why, one thing I don't think I explicitly mentioned, but I think it's worth talking about, right? I think you're all aware that viral genomes mutate at a much higher rate than, let's say, the genomes inside of our bodies, right? I think you already intuitively know that. There are many reasons why. One is simply because a virus particle, that's carrying around its genome in it doesn't have any DNA repair machinery in it, right? Our cells have DNA repair machinery. And as we discuss DNA repair later in the semester, we'll realize, right, that's a big part of how our cells normally fight cancer, right? So these viruses can't fix their broken DNA or RNA. And number two, these viruses, if you sneeze it out or cough it out or whatever, and it's floating through the air, even in that short time that it's floating through the air before somebody breathes it in, right, it's being bombarded by UV radiation, right? And UV radiation, as we will study later, of course, damages DNA, right? Even our cells in our body are subject to UV radiation damage. But of course, that's when we have something like 20 or 30 layers of keratinized skin protecting our cells underneath from UV radiation, right? And yet we still get mutations. So you can imagine a little virus particle that is nothing more than practically naked nucleic acid surrounded by a little cell membrane, right? A little capsule, right? And if that's floating around in the air with the sun shining down on it, oh boy, right? It's bound to get mutations, right? Okay. So we learned that in the case of C-sarc becoming V-sarc, it was due to a mutation that prematurely truncates the chicken sarc gene, so it no longer has the C-terminal, and that led us into our first sort of discussion about how it is that protein structure is important for determining function, like on and off, of different important proto-oncogenic proteins, right? So here again, remember sarc, remember the example we looked at is its C-terminal domain is usually kept sort of wrapped around itself because of the SH2 domain in sarc that can bind to phosphorylated tyrosine at 530. And when that phosphorylation is removed, right, the whole protein unfolds, and that's then what exposes the catalytic or kinase domain of sarc. Right? In a couple more review slides, we'll remember the importance of these other domains that make them very highly conserved and used throughout evolution in many proteins, right? The SH2 and SH3 domains. Right? Remember that an important thing to keep in mind is all of these proto-oncogenes that we've studied, things like RAS, like sarc, whatever, we think of them as being sort of supervisory types of proteins that signal into the cell. And importantly, sort of the big boss proteins that control them are the proteins that are involved in receiving extracellular signals, right, through a multitude of different types of receptors that bind to different things that are all little ligands, right? Remember, ligand just means a small little molecule that a particular receptor expects to bind to. And they can be free-floating types of ligands, or they can be things that are just found in the surrounding extracellular matrix. Right? So receptor tyrosine kinases are a very important group of these when it comes to cancer. Remember that an RTK, like this one here, this could be, for example, an epidermal growth factor receptor. Usually, they are expressed in limited numbers on the surface of the cell, right? So these are just kind of floating around as monomers, floating around. When something like the ligand comes along and arrives at the cell, in this case, an epidermal growth factor molecule, it will encourage two of these monomers to come together. And then why are these called RTKs, right? They are receptors that are tyrosine kinases. So they have these tyrosine kinase type domains in them, right? That are very similar to the tyrosine kinase domain of Sark. And then these proceed, the two monomers proceed to transphosphorylate each other on particular tyrosine residues. Remember, this can be kind of confusing because the actual details of one strand phosphorylating the other is happening in trans, right? But because this is happening in a single activated now dimer receptor, you can also think of it as the receptor is phosphorylating itself, right? So that's why we can say it's autophosphorylation. So why is this important? Remember that tyrosine phosphorylation, right, only represents a minuscule percentage of the overall numbers of phosphorylating protein events happening inside of a living cell at any given time, right? Something like 99% of all protein phosphorylation events that are happening right now inside of your cells involve phosphates being added to serines and threonines. Tyrosines, right, are limited to only something like less than 1% of the phosphorylation events going on, right? So I encouraged you to think about, evolutionarily, right, why is that important, right? Why would Mother Nature reserve tyrosine phosphorylation for only a small, teeny tiny subset of intracellular signaling pathways, right? So here you have a PDGF receptor, and we learned that it has certain tyrosines on it, just like any receptor tyrosine kinase that can be phosphorylated when it's activated. Right here, it's just showing one of the monomers, right? But remember, this activated one, maybe it's better if we had two blue strands next to each other so that you could appreciate that the strands have phosphorylated each other. Anyhow, we said that if you look at these phosphates, right, they are in key amino acid sequences, right? For example, this one here, 579 through 581, those are, of course, the amino acid positions, right? 579, there's a Y, 581, there's a Y, but then in these other positions, there's an isoleucine and a valine. And we said when they are phosphorylated, that's how Sark can recognize this. And Sark uses its SH2 domain. The SH2 domain has this structure where it's two sort of alpha helical structures and then this more sort of beta pleated sheet type of structure in the middle. And it creates two pockets that fit around these two phosphates. Importantly, it'll only fit around them properly because the amino acid sequence of these other parts, the green and the red over here, right, are also forming the right shape to be able to accommodate the isoleucine and the valine. Right? So that's what makes this particular YIYV amino acid sequence, when the phosphates groups get added there, right, that's specific to Sark SH2 domain. Right? And so then you can appreciate how other variations in this amino acid sequence create these phosphotyrosine sites that are recognized by other proteins, right? Also utilizing things like an SH2 domain, but their version of the SH2 domain, right? That might have variations in their amino acids in the green or red part that make it distinctly something like the SH2 domain of PI3 kinase that would recognize YMDM, right? And recognize that phosphate there, for example. Okay. So again, remember that these SH domains, they're called SH domains because they were first identified in Sark, right? That's why our whole story about starting with Sark this semester and that story about the Rouse chicken virus and stuff like that, right? It wasn't meant to just be a funny kind of analogy or history lesson. It's important because Sark became sort of one of the first oncogenes that researchers focused on to understand what was happening because it was identified as the genetic sequence being carried around in Rous sarcoma virus, right? And then, of course, later, they made that shocking discovery, right, that SARC actually is present already in the genome, right? That was pretty shocking at the time, right? That this wasn't something that was made up inside of the chicken virus. It was actually taken from the normal genome, right? And so when they studied SARC, that's when they realized it had these domains. And then later on, as they looked at other proteins that are also involved in recognizing phosphotyrosines, they realized, oh, those proteins have these conserved amino acid sequence in them as well that match the SH2 domain, the SH3 domain, right? So many tyrosine kinases have SH2, SH3 domains. We said that other types of proteins, like GTPase activating proteins, they are not necessarily kinases, right? But they do have some sort of catalytic site. In this case, something that's going to help another protein like RAS activate. But they use SH2 and SH3 domains as well, right, to get to the right location. Phospholipase C that we studied in our last lecture when we looked at PI3 kinase, right? And certain adapter proteins, which look very modest, right, and very sort of innocent. They are nothing more than strings of SH2 and SH3 domains. They don't even have any kind of enzymatic catalytic site on their own. So you might think like, wow, this is weird. It's like a little scrap of something. It can't be that important. But what we've learned, these are very important because they serve as the adapters that bridge other molecules together to bring things all to the same spot, right? And what is that spot that we're talking about? Well, so far in our discussion of receptor tyrosine kinases on the outside of the membrane receiving mitogenic signals, we are talking about the inner surface of the cell membrane, right? So things like RAS, for example, that's already kind of tethered to the inner surface of the membrane, but it's just minding its own business, hanging out there because RAS is normally bound to GDP, right? But then if you have proteins like SOS1, which is a guanosine exchange factor, it will exchange the GDP that RAS has and swap it out for GTP, right? And then when GTP is in the RAS, then the RAS conformation changes and RAS is now the right shape to begin to bind to other cytosolic protein partners that would have just been up until now floating around, right? Bumping off of RAS, not really binding, right? But now that RAS is the right shape, if some of these partners are close by, they will then bind to RAS. And again, how do those partners, right, if they're just floating around in the cytoplasm, how does your cell actually encourage this RAS signaling pathway to start? Well, one of the best ways of encouraging a whole soup or a whole mess of proteins to be able to carry out a specific set of protein-protein interactions in a chain is simply by preferentially localizing those proteins that you want to increase their chance of interacting, get them all into the same place, right? And that's what those like SH2, SH3 domains are all about, right? So RAS, remember, can signal through three main pathways, the RAF pathway, the RAL-GDS pathway, and the PI3 kinase pathway. So we looked at the RAF-MEK pathway, and we looked at the PI3 kinase pathway. We kind of skipped over the RAL-GDS one for the time being, right? Ultimately, we said that RAS is like a supervisor that's going to then control downstream the actions of certain transcription factor proteins. And those proteins are the ones that actually do the nitty-gritty hands-on work, right? Because these are the ones that are actually docking onto regulatory sites, controlling genes. Okay. So RAS, remember we looked at it more closely and we said that our genome has 3 copies of RAS, right, on 3 different chromosomes. And the very fact that they were first identified in different types of cancer, right, and to this day, right, we know that they contribute to particular types of cancers, but not the other ones that the other RAS versions do, already allows us to understand the importance of how our human genome over evolutionary time, right, is subjected to mutations as well. So originally, there must have been one RAS gene that then got duplicated, right, through some event, gene amplification, duplication event, and ended up on 3 different chromosomes. When that first happened, right? All three of them were probably the identical RAS, right? And maybe having 3 copies of identical RAS was okay, right? Because maybe the cell could still control it. But importantly, what happened was as those 3 different copies of the RAS each started getting random mutations as part of evolution, right? The version of RAS that's made from them now is ever so slightly different, such that the NRAS version, right, only interacts with certain proteins or tends to interact with certain proteins that favor things like neuroblastoma if things go wrong, versus the H compared to the HRAS or the KRAS, that if they go wrong, right, their protein binding partners give rise to Harvey sarcoma or Kirsten sarcomas, right? What does that mean? That means they have different interaction partners as a result of being slightly different shaped versions of RAS. Okay, even though for all practical sake, their functional regions are nearly identical. So that's why we said that when we study things like mutations in key proto-oncogenes like RAS, we have to be careful of how we interpret this data about mutations. Right? We said that in almost all cases, the mutation that you see in an oncogenic RAS, right, that was taken from an aggressive tumor biopsy involves either amino acids 12 or 13, right? And remember, this does not mean that other parts of RAS cannot be mutated, right? Because mutations are random, right? It's just the ones that cause the most harm in terms of derailing RAS's normal protein function are the ones involving amino acid 12 and 13, because remember, these are the ones that play a critical role in how RAS hangs on to GTP, right? And if it can hold on to GTP longer and not give it up, right, not catalyze it back to GDP, then the RAS stays on longer, right? So recall then, so we're kind of, we're going in circles here because I want to always encourage you to think about these things again. So now we have RAS here. RAS is sort of the mid-level supervisor, right? Its big boss is this purple receptor tyrosine kinase over here. And the way the big boss tells RAS what to do is it receives some sort of mitogenic signal. You get the phosphorylation events on the tail of the RTK, right? That leads to the SH2 domains of certain proteins being able to bind to the tail. So now things start coming together at the underside of the cell membrane, including things like SOS, right, which binds to the SH3 domains, or I should say more accurately, the SH3 domains of GRAB2 bind to proline-rich regions in SOS, and that gets this orange SOS molecule up here, right, up here next to RAS, where it can do its guanine exchange factor job of exchanging the GDP to GTP, right? At the same time, you have other proteins like this gap that's brought to this scene here because of its SH2 and SH3 domains, right? Everything's being brought together due to phosphorylated tyrosine or tyrosine phosphorylation, right? Because it's brought now close to RAS, it can go ahead and use its gap domain to encourage RAS. No, don't stick around hanging on to GTP. You go ahead and cleave the GTP because that's what you are. You are a GTP ace, right? And then that RAS will do it and it becomes GDP again, and then RAS switches off, right? We also looked at examples of RAS and how RAS helps bring PI3 kinase to this area as well, right? And PI3 kinase works on these phosphated or phosphorylated inositol groups that are found as part of certain phospholipid molecules on the underside of our membrane, right? So remember, PI3K will take PIP2, phosphatidylinositol phosphate 2, like phosphatidylinositol phospholipid 2, right? Sorry, I'm getting tripped up here, right? And add a third phosphate group on it. So it becomes PIP3, right? And PIP3 is the right shape to attract and bind AKT. And again, in this case, AKT would have just been floating around down here, minding its own business. But if it can bind to PIP3, then it's brought close to the underside of the membrane where these purple phosphatidylinositide dependent kinases, or PDKs, right, go ahead and phosphorylate AKT, activating it, right? And then this phospho-AKT then kicks off mitogenic signals inside of the cell. We also had our first introduction to what we will get to know very well as a tumor suppressor protein. Right? So something like PTEN. PTEN reverses PIP3, takes off that third phosphate, converting it back to PIP2. So in this case, AKT won't be encouraged to come to this area, right? So later, as we talk more and more about tumor suppressors, I think you'll come to appreciate that even though the oncogenes are super important for the story of how cancer happens, so also just as important, and maybe to some extent even more, is the constant role of tumor suppressors like PTEN or P53 or other ones that we'll look at, okay? We looked at Wnt signaling. So Wnt signaling is another example of a important pro-proliferation pathway that in this case has nothing to do with phosphotyrosines being the major starting signal. We said that beta-catenin is an example of a transcription factor that normally, right, if it's allowed to exist in the cell and float around, it will get into the nucleus and it will bind to certain partners like this green TCF-LEF that are already parked on things like certain enhancers or promoters of genes, genes that if they are expressed will then start promoting the cell cycle. Right? We said that normally our cells are constantly making beta-catenin, but in an ironic kind of weird way, all of the beta-catenin, well, not all, but most of it is almost like instantly destroyed. Right? So what typically happens on the left is the beta-catenin is bound up by a complex of proteins that involve APC. Right? And I told you later we will look at APC because the name of it was first identified in colon cancer, right? And GSK3 beta. So when it's in this complex, GS3K beta phosphorylates beta-catenin, resulting in beta-catenin being targeted for degradation by the proteasome. So in that case, the beta-catenin that's constantly being made by our cells is constantly being destroyed. Right? However, if the cell receives a mitogenic signal such as Wnt, right, this red thing here, Wnt binds to the frizzled receptor, an example of a transmembrane protein, multipass transmembrane protein. And what that will do is the activated frizzled receptor will prevent that beta-catenin degradation complex from forming. And as a result, right, the GSK3 beta does not phosphorylate the beta-catenin. The beta-catenin is able to escape being degraded, and that's how beta-catenin goes into the nucleus, right, and starts activating genes, in this case by booting off other repressor proteins that were already on there, keeping the transcription turned off. Right? So then beta-catenin comes down and it can boot off these repressor proteins and encourage the other activating proteins to be able to do their thing. So the take-home message about this was that, many examples of our cells or our physiology involve making things like proteins just to degrade them very quickly. Right? And even though that sounds like a very sort of backwards, inefficient way to do things, the reason why certain proteins are regulated that way is because many times when you need those proteins to work, you need them to go to work pretty quickly, right? And I said, if our cell didn't make beta-catenin all the time, and the beta-catenin gene was just always sitting there waiting to be transcribed, right? And then your cell needed, because it receives these mitogenic signals, right? It needs to signal through beta-catenin, right? It could take hours and hours then for enough beta-catenin to be made, right? From that one copy of the gene. And maybe the cell doesn't have hours and hours, right? Because it's trying to respond to different types of signals. So we evolved to produce a lot of beta-catenin, right? And just degrade it so that if we do need it to work, we just simply don't degrade it, right? We don't degrade it like this. So if the Wnt signal arrives very quickly, right, the existing beta-catenin is spared from degradation and goes to work. Right? I think that's fascinating. It's an interesting way of thinking about different mechanisms of how our cells can control their proteins, right, to be very fast acting if needed, even though it sounds very inefficient and wasteful. Okay, this is the last slide for our review. So to kind of put this together, right, we looked at examples of how things like integrin signaling is important for the function of epithelial cells, right? So an example of a non-receptor tyrosine kinase protein that is important for controlling things like potential oncogenic pathways are these integrins, right? And we said that their job is to bind to components of the extracellular matrix. So different things like fibrous proteins that are part of the ECM surrounding the cell. Or more specifically, when we're talking about epithelial cells, we can pretend that these fibrous proteins are part of that basement membrane that the epithelial cell must remain anchored to in order for it to get its proper signals that it's in its happy place and should be doing its fully differentiated epithelial cell jobs, right? So how does that happen? It happens because the integrins on one end have the right shape to bind extracellular or basement membrane proteins, right? On the other side of the cytoplasm of the cell, this gray thing, right? The tails of these integrins interact with various proteins that are sort of part of the cytoskeleton of the cell, right? So many of these include some of the proto-oncogenes that we've been studying, like SRC, PI3 kinase, RAS, right? Many of them are just caught up in this. So when these integrins contact a spot of extracellular matrix, right? In other words, think of the whole surface here of the cell. It's represented by this thick gray line, right? But imagine this is only one little part of it, one teeny tiny spot on it, because the whole surface of the membrane, it's kind of wavy. You can imagine it as being wavy, right? So only certain parts of it, like this spot on the slide here, would be touching the basement membrane. And so in the spot, you get more and more integrins that happen to bind to the ECM, all being physically kind of grouped together or brought together, right? So as they get brought together by these navy blue arrows showing the movement, physical movement of the receptors, then on the other end, the cytoplasmic end, they're also drawing together all the stuff that they're hooked onto, right? Like the cytoskeleton. So this can contribute to things like whether the cytoskeleton, right, is properly structured and helping the cell stay anchored there, right? You can imagine what would happen then if you lose contact with the extracellular matrix. The cytoskeleton might begin to disorganize, right? And at the same time, you also get increased interaction between the different types of signaling proteins that are sort of hitching a ride on the cytoskeleton, if you want to think about that, right? So all of these proteins get brought together as well, so that things like interactions between SH2, SH3 domains can be encouraged to happen. If you had, for example, in the middle of this slide, even though we don't have it in the slide, imagine, right, you had a big receptor tyrosine kinase right in the middle of this, right, surrounded by these integrins, right? Then you can imagine then that perhaps, right, things like these GRAB2, PI3 kinase, SRC, RAS, right, now would be brought close together to where these phosphotyrosines on the receptor tyrosine tail might be in this image. Okay, all right, so that's it for our review. So I'm happy to open it up to any questions that any of you might have. So let me stop the recording